The Society's 2383rd meeting in the 146th year since its founding in 1871. Good evening, everyone.
Good evening.
My name is Larry Milstein. I am the president of PSW, the oldest scientific society of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members and guests to tonight's lecture by Gerald Joyce in the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C., including our members and friends around the world who are watching the live stream of tonight's meeting on PSW Science's YouTube channel. We'll be very uncooperative computers. We will begin with a few announcements, followed by a reading of the minutes of the 2382nd meeting. We will then turn to this evening's lecture, followed by a question and answer period. Thereafter, I will present a small thank you gift to our speaker, make a few closing announcements, and then adjourn the meeting to the social hour. First, please join me in thanking the sponsors of the Fall 2017 and Spring 2018 lecture series, the Policy Studies Organization, in cooperation with the American Public University. Well, it's certainly on here, only over there. Everything's over here. There's a gremlin in the switcher. Well, I was supposed to delete that slide too. Okay. The Policy Studies Organization, in cooperation with the American Public University, and a generous sponsor who is asked to remain anonymous. I am pleased to announce that the following new members have been elected. Prachi Vakaria, a transportation consultant with the World Bank Group, particularly interested in women in science, who learned of PSW through a friend at the U.S. Naval Observatory. Barbara Colton, a science journalist and a professor of journalism, formerly with Nature, the Nature Publishing Group in Science, interested in biomedical research, medicine, health care policy, and research policy, who learned of PSW through PSW member Jay Kaplan. Emily Major, a student at Bishop O'Connell High School, interested in chemistry and biochemistry, who comes to PSW through her dad, Millen White partner Scott Major. And Diego Toro, a professor of mechanical engineering at Catholic University, interested in acoustics and acoustical imaging, who learned of PSW from a friend who is a member. And tonight's speaker, Gerald Joyce, whose interest will be obvious from tonight's proceedings, at least in part. Please join me in welcoming them to the Society. If any of our new members are here tonight, including those who were announced at the last couple of meetings, please see me to pick up your reprint of Volume 1 of the PSW Bulletin, where you can learn something about PSW's founders and, importantly, why they chose the name they did. Recording Secretary James Heelan will now read the minutes of the previous meeting's lecture on Proving That Computer Systems Do What They Are Supposed to Do, given by Byron Cook of Amazon Web Services. James.
Good evening. At the Cosmos Club in Washington, D.C. on November 3rd, 2017. Corresponding Secretary Robin Taylor called the 2382nd meeting of the Society to order at 8:01 p.m. Secretary Taylor announced the order of business, announced the evening's lecture would be live streamed on the internet, and welcomed new members. The minutes of the previous meeting were read and approved. President Larry Milstein then introduced the speaker for the evening, Byron Cook. Cook, Senior Principal at Amazon Web Services and Professor of Computer Science at University College London. His lecture was titled, Automatically Proving that Computer Programs Do What They Are Supposed to Do. Dr. Cook began with an overview of mathematical proofs. Proving something in math is using logic to be certain that a system works. Because proofing uses the tools of logical analysis, it can be applied to systems of all types. To prove something mathematically, he said, takes 2 steps. First, developing the argument, and second, double-checking the argument. From thousands of years ago until very recently, this work was done by human hands. But where it used to be necessary, in Dr. Cook's words, to find a smart person to prove the theorem holds, around 2001, computer scientists started writing software to take place of that smart person. Thus, the techniques of mathematical proofs can be used in almost limitless number of contexts. Dr. Cook, for example, has used software to apply mathematical proof techniques to microprocessors, aircraft software, device drivers, and to biological models such as leukemia and diabetes. But, Cook said, there may be limitations to what computers can do. For example, he said, new students in computer science are invariably introduced to the halting problem. Posed by Alan Turing in the early 20th century, the halting problem asks whether there exists a system that definitively answers whether there exists an infinite loop in any other system. In modern parlance, can you write a program that will predict whether a Windows program will produce an hourglass symbol, or whether an Apple program will show the user a beach ball that never stops spinning? Dr. Cook then described a halting problem proof in computer code. The program code asks, if the program halts, then don't halt. And if the program doesn't halt, then exit, or don't halt. Dr. Cook said the program presents the same kind of problem as the statement, I am a liar, and cannot be solved. But though computer scientists have not solved the halting problem, there remains tremendous commercial application in attempting to identify whether a system contains an infinite loop. For example, Cook says one way to think about cancer is as a termination bug in the regular pathways in cells. As another example, the event handling routine of a computer's mouse device driver should always terminate. When a user moves the mouse, the computer code handling that movement should always do something and then return control back to the computer. Otherwise, the computer will hang. Traditional pre-digital computer methods of proving termination are difficult in practice, and in some cases, impossible in theory. These methods frustrated computer scientists in the pre-digital computer age. But when computer scientists revisited the problem with the power of digital computing, they realized they did not always need to prove termination in every instance for there to be a commercial benefit. To illustrate his point, Cook posited that people fly commercial airlines, even though they are not 100% safe, because the occurrence of crashes is so low that people are comfortable with their chances. So, Cook has sought to recreate, through rough digital facsimiles, certain systems that solve for halting and that produce reasonably reliable but not perfect results. Dr. Cook then described the logic and code for some of his work on device drivers, showing an example of how he used proofs to identify a termination problem in computer code. As he mentioned before, that logic is transferable. For example, he has worked with leukemia researchers to answer the question, when a system is perturbed, does it always go back to a stable state? In sum, Dr. Cook said that computer scientists first realized around 100 years ago that ancient methods of reasoning could be used to analyze modern industrial artifacts. Then, around 50 years ago, computer scientists became disillusioned when they realized the limitations of these methods. But now, due to the pressures of the commercial marketplace, computer scientists have realized that they do not need to academically identify universal truths. Rather, they can begin by working in the field to making tools that perform adequately in niche areas, which can then be generalized and modified for application in other areas. Secretary Taylor then invited questions from the audience. One member asked whether Dr. Cook's techniques could be used to identify errors in the human brain. Perhaps, Cook responded, but his techniques rely on accurate models, so that their application to the human brain would be limited by the accuracy of the brain model. Another member asked whether it was possible to write a program that could identify and repair its own errors. Dr. Cook answered that this question was an area of active research, essentially requiring programs sophisticated enough to search and identify the correct series of inductive questions to identify termination. and a live stream viewer from California asked whether Dr. Cook had applied his techniques to approximations. Yes, Cook said. Representing a very complicated program in a small, finite space can be done by adding non-determinative variables to the program. Proving the correctness of the program when he has added those variables logically means that the correctness of the more complicated original program has also been proven. After the question and answer period, Secretary Taylor thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:21 p.m., Secretary Taylor adjourned the 2,382nd meeting of the society to the social hour. Temperature, 20 C. Weather, overcast. In-person attendance, 85. Respectfully submitted, James Heelan.
Thank you, James. Are there any comments or corrections to the minutes? If not, I will entertain a motion from a member to accept the minutes as read.
So moved.
Do I have a second from a member? All in favor, say aye.
Aye.
All opposed, say nay. Okay. The minutes are accepted as read and will be posted to the website in due course. We now turn to tonight's lecture. It is my pleasure to introduce tonight's speaker, Gerald Joyce. Jerry is a professor at the Salk Institute in the Skirball Center for Chemical Biology and Proteomics. At the same time, he serves as Institute Director of the Genomics Institute of the Novartis Research Foundation. Prior to joining Salk, he was a professor at the Scripps Research Institute. And before that, he did postgraduate medical training at Scripps Mercy Hospital and postdoctoral research at the Salk Institute. Jerry has a long-standing interest in the origins of life and the role of RNA in the early history of life on Earth. His research involves the test tube evolution of functional RNA and DNA molecules and their potential application in clinical diagnostics and in therapeutics. He developed the first self-replicating RNA enzyme capable of exponential growth and evolution in the test tube. And he and his colleagues also developed an RNA enzyme that catalyzes the polymerization and exponential amplification of other RNA molecules. Among other honors, Jerry has been elected to membership in the U.S. National Academy of Sciences, the U.S. National Academy of Medicine, and the American Academy of Arts and Sciences. He is a recipient of the U.S. National Academy of Sciences Award in Molecular Biology, the Hans Siegrist Prize from the University of Bern, and the U.S. National Academy of Sciences Award for Early Earth and Life Sciences. Jerry received a BA from the University of Chicago and both an MD and PhD from a much better institution, the University of California, San Diego, where I got my PhD. Please hold questions for the end, when we'll have a question and answer period, and all of you will have a chance to ask Jerry questions. Please join me in welcoming Jerry to the podium.
All right, let's see if we can make the switch. Just a
As soon
minute
as you
here.
pull it up.
That's me.
Broad.
Oh, we're good. Thank you. Larry, thank you so much for the invitation. It's a pleasure to now, I guess, be a member. So
Yes.
that was a surprise. I didn't realize that speakers get to be members on the spot. What if you don't like to talk? And thank you for the I have to do it again, okay. And thank you for the kind introduction, including the wisecrack about UCSD and my other alma mater, the University of Chicago. So this being the Philosophical Society, and I know that is not taken quite so literally, I thought I would start with a sort of philosophical point, which is, it's kind of a lonely planet, isn't it? It's a beautiful planet. It's all we know, and it's all we know about life. Because as far as we know, we are alone. And so that's kind of sad and kind of humbling and kind of depressing even, you know, that there isn't something else out there, another voice in the void. I don't know. I'm not saying that's what drives my research, but it's kind of sad. So, there's been false alarms, and, every time there's a new interesting extrasolar planet discovered, there's a new article often in the front page, at least in the Science Times, the New York Times, Washington Post. Maybe here, maybe there, Remember, how many people remember this one? Bob knows. Bob Hazen knows this one well. You weren't a co-author, thank goodness. But so we are, well down the road now from the so-called Mars rock, right? This was the big, and I know we have folks from NASA here. I'm not picking on NASA. I'm really speaking to the kind of loneliness and hunger we all have that, that if only there was a voice out there calling to us. President Clinton, in announcing this finding and this paper from the White House podium, said, "Today, rock 84001, that's the Allen Hills meteorite from Antarctica that truly did come from Mars and truly does not bear signs of life." But at the time, he said, it speaks to us. Across all those billions of years and millions of miles, it speaks of the possibility of life. But there's nobody there on this rock, So, so what do we know? Look, we know that we are on this beautiful planet teeming with beautiful life forms. I mean, it's a remarkable thing that's happened on this planet. So maybe rather than just listening to the void, we should focus on what's in front of us, which is a life form that we can, and science and society have come to understand deeply. But the question for tonight is, how does it work? Where did it come from? And really the topic for tonight is Life 2.0. Can we make another one of these things that we call life that we love so much on our planet? We're going to do it on our planet in a test tube, and maybe someone will pick up a signal from space that'll beat us to it, and that would be a lovely day, But we're going to tonight talk about actually trying to make Life 2.0. So let's talk about our life form and how did we get here. Again, a lovely planet. It wasn't so lovely at the beginning. So our planet was formed like 4.56 billion years ago from the circulating dust cloud, the interstellar dust cloud, through a lovely process that goes by the name of orbital gardening. Our planet was sort of gardened up in what was actually a violent series of accretion events leading to the early Earth. Was not a hospitable place for life. If one of these was found by extrasolar planet survey, it would be declared not habitable for life. But eventually our planet cooled down, completed its accretion, developed a stable hydrosphere. I won't go into the specifics. It's not really the topic for tonight, but there is good evidence from what are called detrital zircons that there was a hydrosphere in place by about 4.2 billion years ago. We can now start to think of the planet as actually blue. Again, when you see these stories about extrasolar planets, the artist rendering always colors them blue with a little bit of clouds, But 4.2 billion years ago, we really did have an aqueous planet. In fact, maybe too aqueous. There's a question now whether there really was much landmass at the time, but at least a stable hydrosphere in place, habitable by current way of thinking. And then a kind of dark era. We don't really know. There's a lot of speculation. There's experiments and theorizing about what came before life emerged. This is referred to as the era of prebiotic chemistry. Right? This is the primordial soup. Although it might not have been a soup. It could have been down in the rocks. It could have been at a hydrothermal vent. It could have been a scummy little pond that wasn't very appetizing as a soup. But in any case, chemistry happened, not biological chemistry, just pure chemistry, leading to what then could be the building blocks, setting the table for life. And then, and this is really the focus point for tonight, scientists believe, not universally, but I think it's very broadly accepted, that there was a time in the early history of our planet when life was not based on DNA, genomic material, and proteinaceous catalytic material, as it is now, but instead based on a cousin of DNA, the molecule RNA. This is called the RNA world. Wally Gilbert coined that phrase shortly after the discovery of RNA enzymes by Czech and Altman back in the 80s. The RNA world was a time when there really was a different life form on this planet, a life form based on RNA molecules, RNA genetic material, RNA enzymes that operated on that genetic material and carried out whatever metabolic function existed in that world. Now, I think it's fair to really think of this as a different life form, but not one that is completely disconnected from our life form. So the view is that the RNA world then invented, in a Darwinian sense, our life form. That what used to be RNA genes became DNA genes, what used to be RNA enzymes became taken over for the most part by protein enzymes, and that's the sort of cellular modern life of the last 3.5 or so billion years that we've come to know and love. And after that, well, then it's just history from there. I mean, it's a very interesting, rich history, but that's the engine of Darwinian evolution, churning on our planet, creating diversity. I shouldn't use the word create, exploring the opportunity for diversity to be selectively advantageous, and then those that are more advantageous being enriched over time in a Darwinian process. And just for orientation, here we are. We're right next door to corn. We're just down the street from paramecium in this branch of the tree of life. Okay, so now we're all oriented. This is how we got here, more or less. This is the short version of the story because that's not the topic for tonight. The topic for tonight is the RNA world life form from which our life form emerged. And so the notion is that literally we are the dust of the RNA world. The remnants of the RNA world gave rise to our DNA and protein-based world. And you can see those remnants in our biology, and it didn't have to be that way. The tablet could have been erased, and there might be no remnants of such a prior life gone by, and then it would be pure speculation. But in fact, if you look at the processes of modern biology, you can, in many cases, see the sort of molecular fossils of the RNA world gone by. Many of the most central processes of biology, especially those that concern the processing of biological information, are driven by, moderated by RNA molecules. And I think the key one is the ribosome. So the ribosome, the RNA body, the ribo body, is the entity within all cells of all cellular organisms on Earth, including all of your cells, that makes proteins. Now, if there was an RNA world before a DNA and protein world, if the RNA world evolved into the DNA and protein world, the RNA world had to invent, in a Darwinian sense, DNA, and had to invent proteins. It had to invent a protein synthesizing machinery. And there might not be any trace of that in modern biology, but in fact, the ribosome in your cells is an RNA machine. So here's the crystal structure of the ribosome, the large subunit of the bacterial ribosome. Shown in kind of blue-gray is the RNA part of this machine. Shown in gold, draped about on the outside, is the protein part of this machine. This is an RNA machine. It is still today an RNA enzyme. The site at which the protein is made is right here, the so-called peptidyltransferase site. It's still an RNA machine, this RNA body that makes all of our proteins. So that's a kind of key piece pointing back to an RNA world gone by, but there's many examples if you look in biology. But then we come to this thing, this is the Cold Spring Harbor volume, now in the 4th edition of what's RNA doing out there in biology. But notice on the cover this thing at the center. What's that? What's this RNA with this little circle pointing into itself? This is really what the focus point is for tonight. That's an RNA molecule that makes itself, that catalyzes its own replication, and that therefore brings about its own evolution. That's the dawn of RNA-based life, if such a molecule could be realized historically or experimentally in a life 2.0. Francis Crick has in many cases said this first and said it probably best. Carl Woese said similar things, but Francis, I think, said it best in a review article in Journal of Molecular Biology in 1968. Francis said, possibly the first enzyme, and he put enzyme in quotes because he didn't mean a protein. Francis said, possibly the first enzyme was an RNA molecule with RNA replicase properties. Richard Dawkins in The Selfish Gene said, we will call it the replicator. Now, he capitalized replicator because I think he was making a political point here. It had the extraordinary property of being able to create, I think he used the word create because he was making a political point here, but he's really talking about the same thing, a molecule that can catalyze its own replication and transfer of information from its progeny, thereby enabling Darwinian evolution. All right, so we want one of those. We want to dig one out of the ancient Earth, probably unlikely because RNA is a fragile molecule, relatively speaking, or make one. So what is it we're trying to make in Life 2.0? We want to have an RNA molecule that is an enzyme that catalyzes the production of RNA molecules like itself. So if we have an RNA molecule, which is a string of letters, just like DNA, the letters are slightly different in RNA, it's U instead of T. But imagine there's a string of RNA letters. They're going to be copied to make many copies of that parental string. Give or take the occasional mutation, because these are processes that occur at temperature. They're not going to be perfect. Mutations are going to happen. That's just a bumper sticker. But these are not just strings of RNA. These are molecules that, by virtue of the particular sequence of letters in the string, fold into a shape, a secondary and tertiary structure, and by virtue of that shape, if it's the replicator molecule, if it's the first enzyme that Francis talked about, the replicator that Dawkins talked about, then that folded molecule is the catalyst to bring about the production of more copies of parental to progeny RNAs. So we want one of those. Where are we going to find one? It'd be really cool if there was one of those in biology still. Even if it wasn't doing exactly the same thing in biology still, but maybe a virus was using it to do something for its own sake. No one's found such a molecule in biology, but maybe we can make one. And why do we want such a molecule? Because, as I've already said, once you have a molecule that can bring about this process, Darwinian evolution happens, right? Because if you start with parental molecules that become amplified to make a larger number of progeny molecules, and mutations that occur spontaneously introduce variation among that population of progeny molecules, some subset of that population are going to be more fit, shown in yellow here, compared to their brethren. More fit means better able to persist, copied more rapidly, degraded less rapidly, able to exhibit a function that enables them to be copied more rapidly or degraded less rapidly. The more fit are the ones that preferentially survive, are selected, and become the parents for the next round of amplification, mutation, and selection. So that's Darwinian evolution. And it's just a chemical phenomenon if only you had a molecule that could do this. And how do you find such a molecule if it doesn't exist? I mean, it would be nice if you could just write a computer program and it would design the molecule for you, but that's not going to happen in my lifetime. So instead, we turn to Darwinian evolution itself as a technology, as a process to evolve the molecule, starting from random sequence RNA molecules, and discover those that can do something like this process. So how do we do that? Now, I'm not going to go into the details of the technology. This is a technology that was started back in the late 80s, early 90s. Suffice to say, it's a technology. It's very broadly practiced, and it's used to make all kinds of interesting RNA molecules and other molecules, so-called test tube evolution or directed evolution. But I want to explain how to find a molecule that can take a parental strand of RNA, what's called a template, and bind to that template building blocks of RNA and join them together in an RNA catalyzed reaction, where this piece will become joined to that piece through particular chemistry. I'm not going to go into the chemistry detail, but this is a chemistry that is seen in biology today. Could have been a different chemistry on the early Earth to bring about this joining reaction. Well, how are we going to find a molecule that does that? Using Darwinian evolution, we insert into the template a population of random sequence RNA molecules, just randomly chosen, but 10 to the 15th different randomly chosen sequences. And then we connect the template to one of the two substrates, and we place at the end of the other substrate and at the end of the random sequence containing template molecule little handles. And if and only if, by virtue of the particular sequence that's in this random region, the joining reaction happens, if and only if that happens, will the product molecule have both of these little handles on it. If it doesn't perform the reaction, one handle's on this piece, the other handle's on that piece. But if it performs the reaction, both handles are on the same piece. Those handles are grabbed by an amplification process called PCR, polymerase chain reaction. If and only if the joining reaction occurs, can, through biotechnology, one then make copies of the joined molecules. And remember, you're only making copies then of those sequences that promoted the joining of the molecules. All right, so that's a way to select molecules that bring about this joining reaction, introduce random mutations during the copying process, amplify those that were selected, repeat, repeat, repeat. That's the Darwinian recipe. And that works, just like that works for life on Earth, that works for molecules in the test tube. What once was random sequences turns into, through Darwinian processes, molecules that can bring about that reaction and have a particular sequence, secondary and tertiary structure. So here's an example of one of these RNA joining molecules. Here are two pieces of RNA called A and B. Here's the joining site. And here is what used to be random sequence, but now is a particular sequence of 62 nucleotides, E for enzyme, that binds those two substrates, places them next to each other, and catalyzes the joining reaction with a great rate enhancement compared to the spontaneous reaction, and what's called a Michaelis-Menten constant for the concentration of substrates necessary to support that reaction. Okay, so this happened a while ago, and that's possible. So one can make an RNA enzyme that catalyzes the templated joining of RNA molecules in the test tube, just starting from random sequence. Well, okay, but we need to have replication and evolution. This is just a joining reaction. Well, first of all, just a bit more about this molecule. It turns out, although you see a particular sequence of letters, most of this composition of this molecule is generic. So the part of the molecule that binds the two substrates is just a template that binds whatever is a matching complementary sequence in the Watson-Crick sense. That's true for substrate A and substrate B. What makes this an enzyme, though, is having this particular sort of three-stem structure. It's what's called a three-way junction, stem, stem, stem. And then these particular nucleotides, these particular letters, form the catalytic center of this enzyme. Okay, so now here's a kind of trick, I suppose, that we played. If you have an enzyme like this, it can join together any two substrates that match via the template sequence and have this particular 6 letters at the front end. What happens if A and B, when joined together, make another enzyme? All right, so here's an enzyme with 2 binding regions, but now this region is going to bind another A, and it's going to bind an A substrate, and this region is going to bind a B substrate, place them next to each other, join them together, but the product, if you read from end to end, of the enzyme and the 2 substrates once joined is another enzyme. All right, so formally, A plus B catalyzed by E would make another E. Now, the way we actually do this is we use 2 enzymes. We use kind of a Watson enzyme and a Crick enzyme. Right? So the Watson enzyme, let's call it E, binds the 2 pieces, we'll call them A prime and B prime, and joins them together not to make an identical enzyme, but to make an enzyme that is complementary in the region of interaction. So that's the way information is going to be passed from Watson to the 2 building blocks that become Crick. Then once Crick is formed, then Crick binds the 2 pieces of Watson, joins them together in the same way, and makes a new Watson. All right, so Watson makes Crick, Crick makes Watson, but the most important thing here is then information is passing back and forth from E to E prime, and E prime back to E. So it works like this. Here's an enzyme, here's Watson, diagrammatically, that binds 2 substrates to make this trimolecular complex. Meanwhile, Crick doing the same thing. Chemistry happens. This is the RNA catalyzed reaction that then causes E prime to make E and E to make E prime. And then those 2 pieces come apart. 1 makes 2, 2 makes 4, 4 makes 8, and so on. I'm, of course, leaving a lot of the details out, but this is what happened. Of course, if they didn't fall apart and start the new cycle, it would grind to a halt. But through test tube evolution, we evolved these molecules so that they would continue to just keep doubling. All right, so here's some real data. So now we start with a little seed concentration of the parts of Watson and the parts of Crick. So 5 micromolar A and B and A prime and B prime, and a little input amount of the enzymes E and E prime. And you see that they expand exponentially over time, amplifying more than 100-fold or so after about 6 hours, and then it levels off. Now, why does it level off? Well, because the concentration of building blocks is 250 times more than the starting concentration of enzyme. Once there's a couple 100-fold amplification of the starting amount of enzyme, there's no more building blocks. So it runs out of food. But it grows exponentially until it runs out of food. Now here's how we can keep it going. This is what's called a serial transfer experiment, first pioneered by Saul Spiegelman just over 50 years ago, using viral evolution in the test tube. Here's the idea. We take a test tube and we put in it building blocks of A, B, A prime and B prime, and a little magnesium salt. It does require magnesium salt, and water. That's it. That's all that's in the test tube. Then we put in the test tube a little starter culture of E and E prime, and we let them amplify each other, cross-catalytic amplification, 100-fold so amplification, until they start running out of building blocks. Then we take a small aliquot out of this test tube, say 1 microliter out of the 100 microliter volume that's in the test tube, and we transfer it to a fresh test tube that has a fresh supply of building blocks and magnesium and water. And so on, and so on, and so on. So in each test tube, the molecules have a chance to amplify 100-fold, and then are diluted 100 to 1. And if they don't amplify 100-fold, over time they'll be diluted to nothingness. And if they amplify more than 100-fold, they will grow in numbers. But they're all trying, I shouldn't say trying, they all are competing for the opportunity to do so based on a limited supply of building blocks. So here's again real data from a real experiment that was run over a period of about 37 hours. We call this the Google experiment, not because it was sponsored by Google, but because it's achieved 10 to the 100th-fold amplification, Google, like Googleplex, 10 to the 100th-fold amplification in 37 and a half hours. Okay, so wow, RNA can just do this. It can just keep replicating like crazy. And remember, there's information going back and forth. But now we come to the limitation of the system, why this isn't what we're really after. It's taught us a lot about test tube evolution, about replication dynamics, about exponential growth, and even about Darwinian evolution, because, as I've kept saying, there's a region of the Watson and Crick enzymes that are just Watson-Crick pairing, that are just base pairing, and information is passed back and forth in those regions, and we can use those genes, those genetic loci, to encode traits, functional traits, within the enzymes. But those regions are small. They only contain 6 or 7 nucleotides each. And each nucleotide is worth 2 bits, right? Because there's 4 letters, so it takes 2 bits to encode specifically a particular nucleotide. So if we have a dozen letters to play with, we've only got 24 bits. So it's not a 2-bit system, but it's a 24-bit system. And so you cannot evolve really interesting, complex new functions. You can evolve variants of the enzyme. Remember, the core sequence has to stay the same because that's what makes the engine turn. And we've done experiments like that. But in the end, this has a very limited capacity to evolve. It isn't pluripotential with respect to evolutionary opportunity like life on Earth is. So it's caused us to re-examine, what does it take to not just achieve exponential amplification, self-replication, Darwinian evolution of molecules, but truly inventive, self-sustained Darwinian evolution of molecules. It's forced to take really kind of a whole different perspective on the problem, and in the remaining time, I'm going to tell you about 2 other vignettes where we try to attack the problem of Life 2.0. The first really did require a different way of looking at things, and I show you something familiar, but I tell you, this is not the White House. I'm not making a political statement here. That is not our White House. Does anybody see why? It's very hard to see why. That is not the White House. You've probably seen many pictures of the White House. Many of you even walked down the street and stood at this very spot, maybe not with the weather so beautiful, but at that very spot. And if you stood at that spot and you saw this, instead of what you'd be seeing, you'd say, that's not the White House. Anybody get it? It's hard to see because the White House is so perfectly symmetric, except for some of the spy stuff up on the roof, anti-spy stuff, up on the roof. You'd have to know which way the wind was blowing. But if you knew the shrubbery, if you knew the trees, you would know that's not the White House. That's the enantiomer of the White House. That's the mirror image of the White House. That's the White House. Okay? And of course, if you got up close, you would find all kinds of asymmetries. The West Wing would be where the East Wing was, and vice versa, right? But we're so tuned to thinking symmetrically, we don't notice that the trees are asymmetric. And so what I showed you before wasn't the White House. It's a bizarro world when it was that previous picture. This is the White House we want. Okay. So too with molecules. So when I was telling you about this self-replicating molecule, self-evolving molecule, I never told you what hand it was. So RNA molecules, like all biological macromolecules, come in a right-hand and a left-hand version. It turns out that in the sugars of biology, in the sugar ribose of RNA or deoxyribose of DNA or glucose in metabolism or et cetera, it's a right-handed sugar. So all the data I was showing you before, how did you know I was talking about a right-handed RNA? I could have been talking about left-handed RNA. Nothing would have been different, right? If I had left-handed food, left-handed A and B, and a starting amount of left-handed E, then left-handed E would make more left-handed E, just like right-handed would have. In fact, that's true. So if you synthesize the enantiomeric version of the replicator, it can't help but replicate if it's given its corresponding mirror image food. They can't use each other's food, but of course, what happens in the mirror is identical in a physical-chemical sense. So either D or L replicators, right hand or left hand, do the same thing. But here's the point of departure. It's like, well, okay, so there's these 2 worlds on the other side of the mirror, but if we think about the early history of life, the mirror hadn't been broken yet because life hadn't broken the mirror. If it's just chemistry, who's to say it's left or right? And what happens if you put the left hand and the right hand together in the same place at the same time? How do they shake with each other? Well, just like handshakes, right hand to left handed handshakes don't go very well. You can kind of kludge it, but it doesn't work very well. So too with RNA molecules. So it turns out the rules of Watson-Crick pairing only hold when the hands are the same on the template strand and the complementary strand. You cannot, between 2 different hands of RNA, say a right-handed template and a left-handed complement, you can't make more than 1 base pair in a row. You can't make consecutive base pairs between the opposite hands of RNA. But on the primitive Earth, maybe there were opposite hands of RNA. Maybe that's a clue to a different way you could get this whole thing rolling. What if we put left hand and right hand together and asked them to cooperate in a two-handed process to make a self-replicating evolving system? So we looked into that problem. We began by just asking a more fundamental question: can you play across the mirror? And I'll show you just one example of that, although there's several published cases on this now. So we took a very well-known piece of RNA from biology. This is the so-called TAR regulatory element from HIV-1 virus, retrovirus. And we made this not in the biological right-handed D sense, but in the mirror image L sense. It doesn't exist like that in biology. It exists as D. But we made it as L. And we immobilized it on a solid surface. And we provided a very large population of 10 to the 15th different random D RNA sequences, right-handed RNA sequences flanked by primer binding sites for amplification. We made 10 to the 15th of these D molecules and gave them the opportunity to bind to the L target. But remember, Watson-Crick's off the table because you got a D player and an L player, so they're not going to base pair with each other. How are they going to shake hands when the hands are of opposite sense? Well, we let evolution figure this out, and evolution found motifs. Here's a particular example that we then minimized down to this particular sequence of D RNA binds to this particular target sequence of lRNA, and it does it quite well. Sub-nanomolar binding affinity, so this is the D binding to L. If we flip the mirror, it's the same, has to be the same. L binds to D with roughly the same affinity. There are some slight differences in purity having to do with the quality of the building blocks that make lRNA, that we obtain the building blocks commercially, but as you can see, it's essentially the same affinity. And here's how they talk to each other. They don't do a Watson-Crick handshake. They do a three-dimensional handshake. This is a footprinting analysis to show the points of contact between the two molecules in what's called a protection assay. I won't go through it in detail, but bottom line is the brighter the red, the more the contact between these two players. And so what happens is the grabber molecule uses this central kind of loop structure to grab the end of the target molecule and achieves that binding interaction. Okay, so D and L can talk to each other, and in a kind of interesting way, because although Watson-Crick is a good way to pass information, it may not be a good way to make functional handshakes, because there's a kind of tyranny of Watson-Crickiness, which is it's so easy to get binding affinity just by dialing in base pairs that it dominates the sort of functional landscape. If instead you let the molecules seek the best functional solution rather than stability solution, let's see what we can get. All right, so let's make an enzyme for replicating things that does it cross-chirally, where we're going to ask the two hands to work together. So what we did was we made a target out of L. So here again, a template, two pieces of RNA. So this is the template. Here's our two substrates, same chemistry, but now it's L, left hand. We immobilize it on a solid support, and we connect the template to 70 random sequence D RNAs, flanked by primer binding sites. Now, if any one of those D sequences can bring about that joining reaction, then the molecule that did so becomes joined to the solid support and can be recovered. Those that didn't do so are discarded. Right? So that's the selection here, is in a cross-chiral way, bring about this joining reaction. If you do, you survive. We will make amplified copies, mutational errors, and repeat. After 10 rounds of this, out pops a cross-chiral catalyst, a D catalyst. We then added 30 more random nucleotides, did some more evolution, changed the format because we wanted it to operate as a separate molecule, not one that only operates connected. So we changed the connectivity, now linking it not to the back to the end of the template, but to the end of one of the two substrates, did more rounds of evolution, trimmed that molecule down, and here we go. All right, so now we have an 83 nucleotide particular sequence and structure of a dRNA molecule, right-handed RNA molecule, that's function is to bring about the templated joining of lRNA molecules. All right, now what does this look like? We'll put a fluorescent label on one of the two substrates. Let's put a red label, literally, a red label for the L. And you see that if we take the D enzyme and ask it to join this short L substrate to this unlabeled second substrate, it'll make a larger product. So the higher up, the larger the molecule. So starting, and then as the reaction proceeds, it generates the product with a nice rate and Michaelis constant. If we flip the mirror, of course, it behaves the same. Now in green, fluorescein, here's the L enzyme putting together the reaction for the D substrate to make the D product. And we can put them both in the same pot at the same time. D makes L makes D. So this might be a different way to think about replication that literally could be D makes L makes D. What would that look like? Well, first of all, this molecule's not good enough yet. It's just doing a single joining reaction. We need to do more evolution to train it to do multiple successive joining reactions. So now we have a separate template. We have lots of little substrates. So here's the first starter substrate, what's called a primer. And here are a variety of what are called trinucleotides. So those are 3-letter substrates, each with a reactive front end and back end, until you get to the end of the line, and then you get yourself bound to a solid support and selected. So if you're not to personify it, but I think we often in the lab put ourselves in the position of the molecules, usually to try to think, what's the nastiest thing they could do to not do what we want them to do? All right, that's what it's actually like in the lab. It's like squeezing a balloon I mean, if anyone here is a horticulturist or is, I don't know, breeds like special rabbits or, cats or something, you know what it's like, right? So it's like, well, that has all the right features except, God, the nose is kind of crinkly. I think I'll take it though. Maybe the nose will get better in a few generations. Oh, it didn't. It just got worse. Okay, let me go back. So that's what we do when we breed molecules. It's molecular husbandry, so to speak. Anyway, so we try to outthink the molecules, and here what we're asking them to do is to join all these little building blocks together in order to win. And if they do so, they will be selected, amplified, mutated, and repeat. So this drove the enzyme to become much better. Now here's a long template, a primer, and a whole series of trinucleotides that it starts with the 13-nucleotide primer and adds 1, 2, 3, 4, just goes right up the ladder. Okay, so now we've built a trinucleotide polymerase through this cross-chiral handshake, through this handshake across the mirror. And that means we can actually do something kind of cool, which is we can take the L enzyme and break it into pieces, and then ask the D enzyme in a templated manner to join all those pieces together. So now here's the D enzyme, starting with the first piece, which is the 7-nucleotide primer. The red boxes are all the little junctions that we put in, and it puts piece after piece together until the D enzyme assembles in a piecewise fashion the L enzyme. Then we can purify the L enzyme and show that it's reactive back the other way. So literally, D is making L chunk by chunk, and L then can make D in a mirror image fashion. I don't know if this is how life started on Earth, and I don't know if this is what's going to get us all the way there in the test tube, but this is one of the avenues that we're pursuing. And of course, now what we're evolving this enzyme to do is add not 3 by 3, but 1 by 1 by 1 by 1. And so we continue to work on this one. I want to end, though, with what I think is our best prospects. So let's go back to the broader goal, a template, a primer, and individual building blocks of RNA, individual letters. They're going to be added in a template-directed fashion. Okay, now we're back on the same side of the mirror for the rest of the talk. So no more, no more weird enantio White Houses. So everything is back on the D side of the mirror. We have a D template, a D primer, and D monomeric building blocks of RNA. And here's another one of those enzymes made out of random sequences. This was work done by David Bartel and Jack Shostak some time ago at Harvard Medical School. And they found out of random 220-mer sequences a particular complex sequence of RNA that brings about that same joining reaction. So here's the template, here's the primer, and here's where the chemistry takes place. It's a pretty nice enzyme. Many groups in the world have been working on this enzyme for a long time because it's always seemed like one of the hot leads. But a long time is a long time. So I'm going to take you all the way up to unpublished work, and I am assured that even though this is being videotaped and simulcast, that I am not doing a public disclosure that would prevent me from publishing this in a peer-reviewed journal. There is nothing proprietary in intellectual property sense, but I'm just going to show you all the way up to last week's data. All right, so that's back in '93. Dave Bartel subsequently showed that you can provide a separate template and primer that can be extended nucleotide by nucleotide in a polymerase-like manner. So not just a joining reaction, but adding several successive letters. They were able to add 14 letters in a row, and later Peter Unruh's group at Simon Fraser University got up to 21 letters in a row. The enzyme's gotten a little bigger. It's picked up one of those random sequence domains that evolved into an enhancement of function for this molecule. This is called the class 1 polymerase. Now we're getting closer, 2011. Phil Holliger's group at the MRC Cambridge found that if you build a little grabber domain at the end of the enzyme, so here's a little sequence tag at the end of the enzyme that binds to the end of the template in a Watson-Crick fashion, just to kind of hang on to the template. It turns out that all previous versions of this molecule were pretty fast, but couldn't really hold on to the template. So they discovered a way to give it proximity, to hang on to the end of the template and extend the primer. And it did very well. For particular sequences, it could add more than 100 nucleotides in a row, 100 letters. But it turned out only for particular very favorable sequences that didn't have any structure to them. So now this is where we came into the game, and now I'm getting you much closer to the present. This is about a year ago now. Dave Horning and myself published a paper in PNAS where through 24 rounds of test tube evolution that I'm not going to describe in detail, we really pushed hard on this molecule to be able to make copies of almost any RNA sequence by challenging it to make copies of almost any RNA sequence. The circles show the mutations that arose during these 24 generations of evolution compared to the previous model. And now we have an enzyme that's pretty fast, much faster than the earlier versions, a couple hundred times faster. But this is the kind of template that had been used previously. It's a template that has mostly what are called pyrimidines, C's and U's, because this enzyme, the previous versions of this enzyme, would choke when they would hit purines, A's and G's. You say, well, that's okay, we'll just copy C's and U's. Here's why that doesn't work for replication. If the template has C's and U's, then the complement is going to have lots of A's and G's. And so you may be able to copy Watson to Crick, but you'll never get back to Watson because you got to be able to eventually copy all the letters. It also, the previous versions couldn't copy regions that had any kind of structure because when it ran into structure, it would stop. And complex molecules have structure. So in evolving the more recent version, last year's version, we pushed very hard to plow through difficult sequences, difficult structures, and ended up with, whoops, go back one. Whoops, that's forward. Able to copy sequence that contain lots of A's and G's in a row, as well as, of course, lots of U's and C's in a row, as well as pretty much any sequence, and even copy through structured regions. And so what this lets us be able to do is actually make complex molecules. So here's a primer, and here's one of these joining enzymes. It's actually a cousin of the replicator molecule I showed you previously. And here's the primer, the starting piece, that is extended by adding individual letters to eventually build an entire RNA enzyme that has structure in it, that has difficult sequences. Here's a whole bunch of U's in a row, which meant the template had a whole bunch of A's in a row, and so on. And then, and this was the picture that I used for the poster that you saw for this lecture, it's now able to copy the copies, right? So here's how this works. We have a template and a primer, and then in a PCR-like fashion, that primer is extended letter by letter to make a complete copy of the template. And then we use that copy, so let's call this one Watson. I've been using that phrasing. So Watson then makes Crick, separate the two strands, then Crick binds a primer that is extended to make Watson, and that brings us back. We call this ribo-PCR because it's like PCR, the polymerase chain reaction, but like the RNA world, it's before there was DNA and protein. So there's no DNA in here, there are no proteins in here. All there is are the template, the primers, the building blocks of RNA, what are called the NTPs, the 4 letters of RNA, and the RNA enzyme, and a little magnesium and buffer. So this is RNA-catalyzed amplification of other RNA molecules, and you see that if there's no starting template, these are temperature cycles, and this is the yield. We achieve 1,000-fold amplification after 24 temperature cycles. Okay, so now we're copying things, and now we're copying complex things. And this was the actual picture that was on the poster. And it behaves, like molecules behave when they have the right properties. Depending on the input amount of template, the lower the input amount of template, the more amplification cycles it takes for the thing to reach the exponential threshold. So this is actual data. This is no template. And then the color code here, the rainbow colors, are increasing template concentrations. And what it's tracking is how many cycles does it take to reach that little threshold line. And that's a log linear relationship because that's what exponential amplification is. It's a log linear, in this case, doubling reaction with each temperature cycle. Okay, so that's good. That's copying and copying the copies and exponential amplification and so on. but it can't copy itself. All right? I showed you it copying another enzyme, but it's a pretty big enzyme, and it struggles in a couple of these regions. So now what are we going to do? Now I'm bringing you to, from here on, unpublished, but one other little bit of published, but mostly unpublished work. Well, one thing we've done is we've taken a divide and conquer strategy, okay? So we take the enzyme, and instead of having it be one long piece, we divide it at 3 different junctions into 4 different pieces. And now we ask the enzyme, the ribozyme, to make each piece individually, but then the pieces can come together non-covalently and assemble into the functional enzyme, all right? So this is unpublished work from Katrina Chung in the lab, grad student in the lab. Here's the whole enzyme as one big piece. Here's the enzyme divided into 3 pieces or divided into 4 pieces, and this is tracking the copying reaction of that enzyme as 1 piece, as 3 pieces, and as 4 pieces. Struggles as 4 pieces, does really well as 3 pieces. So this is one approach. This is our divide and conquer approach. Instead of making this whole enzyme end-to-end, to make fragments and then let the fragments assemble spontaneously to then be the catalyst that makes more fragments. But really what we're focusing on is just making the enzyme better, because we feel like we're pretty close. So here's last year's model that I already showed you, and here's unpublished this year's model, many rounds of evolution later, pressing even harder on sequence generality, overcoming difficult structures, and so on. And now it's to the point it can do some pretty fancy tricks. I would put this as tricks, because it can't yet, spoiler alert here, make itself. But it can make very complex structured RNAs like tRNA. So here's a primer that is extended to complete an entire yeast phenylalanine tRNA. This is last year's model. You can barely see the full-length tRNA. There's a little bit there. It's a fraction of a percent. But here's this year's model making tRNA in a couple percent. And I should say this data is now a few months old. So we're beyond round 38, and we keep going with this molecule. It can also, for the first time, make its own ancestor, because if you remember this part of the story, it started with the Dave Bartel, Jack Shostak ligase ribozyme that then became a polymerase, and then became a polymerase with an attachment, and then became a better one and a better one. But its ancestor, in an evolutionary sense, literally in an evolutionary sense in the test tube, is the class 1 ligase. And we now, for the first time, can at least make grandpa, or great-great-great-grandpa. So the ribozyme, the RNA enzyme, is now extending a primer to actually synthesize the catalytic center of itself. And it couldn't do that last year, but it now can do that this year. So that's kind of where it is. If you have me back, I don't know, in the future, maybe not next year, maybe it'll take more than that, but we hope to keep pushing away on this. So let me come back to the timeline. What I've been focusing on is this period of time, presumed time in the early history of life on Earth, when there was RNA-based life that ruled the day, and DNA and protein-based life was yet to be invented. But if you think about that transition from RNA life to modern DNA and protein-based life, there's, as I've already said, 2 key things to think about. One is RNA has to come to be able to make protein. And we think we know how that happened because we can still see it in the structure of the ribosome. The other is RNA has to make DNA. And you say, well, that's probably easy, right? Because if you can make RNA, you should be able to make DNA. Well, it's not so easy, actually, because chemically DNA is much less reactive than RNA by 10 to 100-fold on a monomer by monomer level. But DNA is also much more stable than RNA. It's 1,000,000 times more stable to spontaneous degradation compared to RNA. That's, in a teleological sense, why your genome is made out of DNA rather than RNA. If your genome was made out of RNA, well, you wouldn't be here because your genome couldn't be more than about 10,000 nucleotides, and you wouldn't be able to be philosophical. You wouldn't be able to be much of anything. Okay? But with DNA, 1,000,000 times more stable, we can have genomes that are 1,000,000, even billions of letters, plus error correction, all those things that happen. So you can see why DNA was selectively advantageous once you got there, but how do you get there? So we thought, well, maybe we can walk there from RNA polymerization to DNA polymerization. So the RNA enzymes I've been telling you about take an RNA template and an RNA primer and extend letter by letter to copy that template for Watson to make Crick and Crick to make Watson. What if instead we have an RNA template and ask the enzyme to make DNA, what's called reverse transcription, as a bridge between RNA information and DNA information? And all previous versions of this enzyme were unable to do this reaction, but our most recent version, and this was just published in eLife last week, or week before last, Our most recent version is now able to do this reaction. So here's the amplification, just a time course, of making RNA on an RNA template, and here's a time course of making DNA on an RNA template. So those two activities are next to each other in evolution space. It makes me think that if there was an RNA world on Earth, getting then to DNA genomes, assuming you had the building blocks of DNA, and that's really a topic in prebiotic chemistry, you could get there. And I won't go through this in detail, but we've challenged it to perform this reverse transcription reaction on a variety of different templates. It likes some better than others. It hasn't been evolved to do this yet. This just was what's called catalytic promiscuity. It's an activity that we were able to get a piece of from what we already had. So this shows full length. Some sequences it copies full length, including long ones that contain all 4 letters. But it's not as good at copying RNA to DNA as it is at copying RNA to RNA. So you can guess what we're doing in the lab right now. We are using test tube evolution to train this molecule to be a better reverse transcriptase. And I think what we're beginning to do is create a divergence, because we are no longer training that lineage to be an RNA polymerase, just to be a reverse transcriptase. So there was this moment of catalytic promiscuity where the two activities overlapped, and now we're optimizing them both from here. Well, of course, once you get to DNA, what good is it if you can't get back, right? So now this is unpublished. If we now have not an RNA template, but a DNA template that we made off of RNA, right? So if Watson was RNA and then made Crick as DNA, we got to get back. We either got to copy the DNA to more DNA and then get back to RNA, or just copy the DNA back to RNA. And very recently, we've shown now that we could do this too. We are better at taking so in all cases here, it's a DNA template for the first time, now copying the building blocks of RNA, or, and not a lot of activity yet, just a few letters being added, DNA making more DNA. So I think of it like this. There was a time, allegedly, 3.6 billion years ago, give or take, a couple 100 million years, when RNA ran the show. This is the RNA world time that we are trying to recreate through technology, where RNA, that's the replicator, right? That's the Francis Crick first enzyme. That's the little symbol that was on the cover of the book volume I showed you, where RNA, its key function is making more of itself. But then we've shown that RNA can reverse transcribe into DNA, catalyzed by RNA. And you can get from DNA back to RNA, and DNA making more DNA. So I don't see that as an obstacle anymore in imagining how that took place, just as if you look at the structure of the ribosome, I don't see that as an obstacle either. I can see, at least in principle, how our life form connects to the RNA world life form that preceded us. All right, that's what I wanted to talk about tonight, other than to tell you to stay tuned. We're listening in here now to see what can answer back. I want to thank the people in my lab who did the work I talked about tonight. Former grad student Tracy Lincoln and former postdoc Michael Robertson worked on the replicator molecule. John Sapansky's now on the faculty at Texas A&M University, and he worked on the cross-chiral enzyme. The right hand makes the left, and the left hand makes the right. And then Eric Mertfield did the most recent versions of that. The last set of experiments I talked about, the best versions of the RNA-dependent RNA polymerase was work of a postdoc, David Horning. Katrina Tung did the divide and conquer work I talked about. And Biswajit Samantha, a former postdoc in the lab, he developed the reverse transcriptase enzyme that was just published a couple weeks ago. And Biswajit is a really interesting character. I've had a lot of wonderful people in my lab over the years, including Biswajit. Biswajit, after leaving my lab, joined a monastery in India, and for the next 5 years does not have access to any electronics while he is a monk in training. So fortunately, we made a deal with his, I'm not sure what the position is called, but with one of the elders in the monastery who does have access to the internet in the library. And through a Gmail account, I was able to send him his paper when it came out the week before last and get a response back that he was glad to see the paper was published. So that's Biswajit for you. Again, thank you for the invitation. It was a real pleasure. I'm happy to take questions.
So we do have time for questions, and we do have a procedure. There are people with microphones in the room. I think they have numbers. Yes? Microphones have numbers? And when you
Yes.
get the microphone, and I point you, please stand up, tell us your name, and tell us if you are a member or you're not a member, and ask a question.
As opposed to making a speech.
It's not working. Can you hear me now? Okay, good. So there are microphones. When you get the microphone and I point to you, stand up, tell us your name, tell us if you remember, and ask a question. So I think, David, you can be first.
Okay. David Rosen, member.
You have to put the microphone close to your mouth.
David Rosen, a lifetime member. At any point, did you consider minerals or self-assembling crystals as as one of your steps towards replicating this DNA? Maybe there's a mineral catalyst or something?
By the way, I wasn't walking away from you. I'm just channeling Bob Hazen. Because Bob has done some of the nicest work, theoretical and experimental, on that opportunity space. Yes, I've considered it. I've read Bob and others' work about this issue. I'm not sure what mineral I need to use right now. I could go into a kind of high-throughput screening of potential minerals and so on. I'd love to be able to do it on first principle. You have a suggestion? Mica.
Manganese oxide with some vanadium, because there was a talk in this building from somebody.
Yeah.
There was a talk a couple of years ago where somebody was showing someone from Israel or something was showing a manganese, this self-replicating manganese oxide crystals with some type of assembly, and I was thinking maybe these are some type of
maybe, but here's how I feel about manganese, because the pK of the metal hydrate of manganese is like 10.2, so manganese and RNA are very unfriendly to each other. So when I talked about RNA being fragile, if we lived in manganese world, your genome, if it was made out of RNA, would be about 50 nucleotides, because manganese is pretty hot for RNA. However, the point's well taken. There may be some mineral that itself can propagate, not in a Darwinian sense, but in a mineralic, phase propagation or something like that, and that maybe the RNA could play off of that, hitch a ride, or play off of particular surfaces, or even help shape surfaces that enable its replication in a Darwinian sense. So there'd be a kind of coevolution between mineral substrate and organic molecule. Very possible to think about that, and Bob and others have written about that. Look, you can see what my game is. It's day-to-day. It's pretty pragmatic in the lab. We know what we have to do. We've got to get about another 10-fold in rate, and just a bit more sequence generality, and whether we have to do the divide and conquer strategy or the end-to-end. Our goal is just to get there, because once we have a ribozyme that can copy itself, then I think we'll see open-ended Darwinian evolution. And then for fun, we'll sprinkle some mineral in there. Hey, at that point, I wouldn't be afraid to sprinkle in some manganese, because now evolution can say, manganese, we know what to do. Hide all the pyrimidines inside stems so they don't get fried by the manganese in a Darwinian sense. So we're just trying to get to a particular goal that I think we can get to by the type of technology we have. But in a broader sense, your point's very well taken.
So, who has the microphone?
I have a microphone. I'm Teresa Fornaro from Carnegie Institution for Science. I'm not a member. Well, in your experiments, you used magnesium chloride solution. So I was wondering whether you studied the possible role of magnesium cations in the amplification.
Yeah, and I have a love-hate relationship with magnesium. So this is how I feel about manganese. Here's how I feel about magnesium. Magnesium also promotes the breakdown of RNA. And if you saw, I always had, like in the bottom right corner of my slides, the actual reaction conditions. It's a lot of magnesium.
Yeah.
So that's a little painful to me, but we don't know in great detail what the magnesium is doing, but I can give you a more general answer. Magnesium supports the secondary and tertiary structure of the molecule. We don't have a crystal structure of our enzyme, but the class 1 ligase catalytic core, Dave Bartel did publish a crystal structure of that, and some of the magnesium ions help nail down that structure. So it helps shield the negative charge on the phosphates and bring them into closer proximity. But there's another role of magnesium that is based on biochemical data. It actually assists in the chemical reaction, in the chemical step. It assists in pulling the proton off of the 3' hydroxyl nucleophile so that it can attack the alpha phosphate of the triphosphate to make the bond. So there's a catalytic role for magnesium and a structural role for magnesium. if we had the nerve, we would say, let's evolve the thing to not need so much magnesium. But we're sort of the molecules, and therefore we are addicted to it right now.
Thanks.
Joel?
Yes. Joe Wilson, member. I had a question, Professor. With regards to the RNA world, not just the ribosome, but perhaps single-strand and double-strand RNA viruses might be indications of it, because isn't the central dogma, it's DNA, RNA replication, but you're suggesting something different. You're suggesting RNA, DNA.
Yeah. So I guess there's kind of two parts of the question. One is, single strand versus double strand, and the other is there some connection to RNA viruses, right? So, even a single strand piece of RNA end to end is largely double stranded because it folds into that. But I think you meant complete double strand. It is possible that in the replication cycle in the RNA world, there was a double strand intermediate. You do have to do the strand separation like we did when we did the ribo-PCR, maybe through heating cycles, some other denaturation cycle, some kind of strand displacement mechanism. I'm agnostic on that. Could have been that there was a full double-strand intermediate. Could have been like, some of the RNA-dependent RNA polymerase replicated viruses that never have a complete double-strand intermediate, like Q beta, a bacteriophage, for example. But now we come to the viruses. Do I think the viruses, were something to do with how life began? And maybe in an inspirational sense, but not in a literal sense. Because, to me, a virus is not alive if I was kind of forced to put on one side of the line or the other, because it doesn't evolve in a self-sustained manner. The virus is a parasite of a cellular entity that provides the machinery to enable the replication, the amplification, mutation, and selection of the virus. It cannot evolve on its own. It can only evolve in the context of a host cell. And so I wouldn't literally say, well, maybe it started with viruses, because literally that's saying maybe it didn't start, because the virus needs the crutch of something that's already going. But if you mean it in a more inspirational sense, a viral-like entity is kind of what we're talking about. There's not much ancillary function here than just making the copies. And of course, notice I didn't talk about cells, or coat molecules. They wouldn't be coat proteins because before proteins, but a sort of RNA shell or a lipid shell or something like that. Maybe that all happened. It's hard to think that this would be completely. Maybe it was on a mineral surface, to hide out from the harsh, broad environment, or to sequester the fruits of your metabolic activity for your own, selective advantage. So I can imagine something inspirationally like a virus, just not literally.
Bob Hershey, I'm a member. Can you extrapolate how long it would take for random steps to come to the steps that you came to?
Yeah, so so from pure randomness to the final answer, that's not what we're talking about. What we're talking about is incremental steps, right? And that's how evolution works. So although we started with random sequence, literally, by making molecules of random sequence, the final molecules just didn't pop up. So, and of course, we have all these generations frozen in the freezer, round by round. They're all sitting in the freezer. We just take a little bit for each next round and freeze the rest. So we've gone back and replayed the tape, and asked what happened at what time. And what happens is, a few mutations kind of get things going, then some more. And you saw when I was showing some of this later versions of the polymerase, those little circles. So here's 13 more mutations that happened over 15 more generations, and here's a dozen more. So they just keep piling on. So and this is a classic creationist argument that people in the field high school science teachers have faced this. It's like, well, how could you have life? It'd be like a 747 just magically appearing on the runway. It's like, well, no, actually, first you developed a technology that made screws, and then there was a technology that made bolts, and then there was a technology in metallurgy, and everything sort of evolved until you could actually put together something complex. But we actually have that entire history. We can see generation by generation. Some mutations confer advantage for a while, and then something else arises that makes what was advantageous no longer advantageous, and it drops out. So there's all this ebb and flow over the many rounds of test tube evolution. I should say it's not that many rounds, right? So it's not like we're using 100 million years of effort here, Now we are, I'm trying to avoid using a deity analogy here, but we are overseeing, we are supervising this process, but we are not Maxwell's demon in there deciding which molecules are winners and losers.
I have a question over here from the guy in the blue, and then Dale, you can go, and then we're going to have a question from Bob Hazen up here.
Oh, good. Yeah.
My name is Mark Cameron. I'm not a member. I'm curious, when you were talking about evolving the RNA, how you were able to make it so that the RNA that did the tasks that you wanted, that was what evolved. If you could sort of go through again and explain.
Yeah, I went through it fast, and there were several different examples, right? So in the general sense, we just have to have a way to separate winners and losers, right? And one way we did that was, if you win, if you do the reaction, then both of your PCR primers are unified. And so you get amplified only if you did the joining. In another case, only if you did the joining do you pick up that little circle with a B in it, stood for biotin. You pick up a biotin moiety that causes you to bind to a streptavidin protein to get pulled away. But it's basically, that's what I say, we're supervising, right, but not micromanaging. So we have 10 to the 15th molecules. Typically, we want something like 0.1% of them or 1% of them to pull it off. If it's more than that, we're not pushing them hard enough. So 99% of the molecules get discarded. 1% of the molecules that perform the reaction under ever more strenuous conditions get grabbed or tagged or flagged or something, segregated. And once they're segregated, we only amplify the segregated ones. So it's just technology.
Dale?
I'm Dale Doucette. I'm a member. You have up here Life 2.0. Do you foresee a 3.0 or maybe even a 4?
Look, even that's aspirational. We're at like 1.01 right now. But yeah, I don't know. That's a kind of slang. I've used that title once before, and it's maybe a little dangerous. Sure. So what I hope is that not just what we're trying to do, but that many labs with many different approaches, some of them not involving RNA, show that chemistry could become biology. Molecules can have history if they can begin to evolve in a Darwinian sense in a self-sustained manner. And whether the substrate's RNA or some silicon-based thing or whatever, we could just keep going. So I hope there's many examples of this. I think once we get to RNA-based Life 2.0, the test tube version of RNA-based life, if it did things like invented a translational machinery, whether it's literally proteins or some other translation machinery to a different functional molecule, that starts to get to 3.0, or maybe that's 2.5. So, I mean, what do we think our life is? Our life form is not 1.0 on this planet, right? And I'm not even sure RNA world was life form 1.0. We don't actually know how life began on this planet. It's very strong evidence that there was an RNA life form before ours, but the RNA life form on our planet could have been 2.0 or even 3.0.
Bob Hazen, Carnegie. One quick question. What's the error rate in these kinds of self-replication? And the second one, have you ever gone back to one of your very early generations, 2 or 3, and tried it over again to see if you converge on the same solution or have a very different solution?
Okay, all right. So question about error rate and question about replaying the tape, but literally, like doing it again. Okay, so error rate. We don't have good error numbers yet on the most recent versions, but what I called last year's model, the 2016 PNAS paper, we did deep sequencing analysis of the molecules, and you got to be careful. You can't just take like the full-length product that it synthesizes, because when it makes an error, it often stops polymerizing. So we analyzed everything, and we found that the error rate is about 3 or 4%, but almost all of those errors are what are called wobble mutations. That's a G pairing with a U. And if you ignore the wobble mutations, which may not be fair to ignore, but if you ignore the wobble mutations, the error rate is about 1% or so. So guess what we're evolving the molecules to do? To stop wobbling so much. So we put wobble penalties on them, and if they wobble, so we're trying to work on that. The replay of the tape, do it again question. Where we are now, we're so deep into this game. We often, in a pragmatic way, we'll back up and start again, right? Or branch and then prune. So we're not running one. When I say we're round 38, it doesn't mean we only did 38 rounds in a row. As I said, it's molecular husbandry. We're doing some hopeful monsters over here, and we're trying this, and we turned the screws too tight. It's like, we were too soft on the molecules. They got lazy. They learned a bad trick. Now we got to unlearn that. We're always kind of creating branches and pruning them. What's harder to say is, let's go start absolutely over. Early days when we first started doing test tube evolution, we did do experiments like that. And the answer we found, and Niles Lehman at Portland State has done this quite a bit more. What we found and what Niles has found too is that for a modest number of generations, and this kind of gets to the question about the accumulation of mutations, there are some things that are sort of there to get. These 2 mutations are going to really take you far, so those get discovered again. But the further you go down evolutionary history, the more it becomes a contingent history. And the more opportunity for a stochastic event that, that didn't happen. Well, because that didn't happen, then none of that happened, and instead this happened, And so that's the kind of thing we see to some extent when we replay the tape, but there's not much mood in the lab to do a bunch of that kind of thing right now. But, I think what's the use of all this? I mean, maybe at the Philosophical Society, I don't even have to raise that question, right? The use is because we're learning something about the fundamental properties of what life is. But what's the use of all this? Well, there is a whole technology of using test tube evolution to make functional RNA molecules for diagnostics, therapeutics. One of our evolved molecules is in clinical trials. I won't even talk about that. But I think in an intellectual sense, what's the use of this? It's a test bed for asking questions about evolution. Right? Now you might say, yeah, but it's a toy system, it's very artificial. Well, but we and others have looked at things like speciation, niche formation, and so on. And when you have a test tube evolution system that is an experimental system, you can play what-if games. So I wish there was more of that. It's very hard to get funding for those kind of questions, but we do a little bit of that on the side.
Al?
Al Ehrlich, I am a member. I was just wondering about the energy balance. When these elements come together to get longer, you're gaining energy.
Yep.
But on the other hand, you're decreasing entropy where you're losing energy.
Yep.
And I was just wondering, what's the balance? And if you need energy, where does it come from? The thermal ambient temperatures?
Yeah, no, good question. So what's the energetic driving force for this? So, we're not at equilibrium. This is an open system. It's being fed. What it's being fed by are the letters. The individual building blocks are what are called nucleoside triphosphates. So you saw on a lot of my slides, there was 3 little letter P's in a row before the letter, right? PPP. That stands for triphosphate. That's a high energy group. It's kinetically almost inert, so it doesn't react spontaneously in water, but if catalyzed, then reacts and is 10 kcal per mole negative delta G0. So each joining reaction has a 10 kcal per mole thermodynamic driving force, and we keep feeding in more building blocks. And to me, you say, well, where did the building blocks come from? Well, from Sigma Aldrich. That's where they came from, because that's where we bought them. So yeah, but where'd they come from on the primitive Earth? Well, that's a prebiotic chemistry question. Yeah, but how can you just use those for free in your system? Because there's no information in the individual building block. The information comes from the particular ordering of the building blocks along a string, and then that information has value or not, and has heritability or not. So, but the answer to your question is it's an open system driven at 10 kcals per mole per bond formation. It's there to get.
I have one more question here.
Kareem Washington, non-member. So I was just wondering, you talked a little bit about tautomeric shifts. And so as you go on with building these molecules, as they do their synthesis, how does that relate to the change or the mutation rate? Since you're going to get bifurcated hydrogen bonds and different types of bonds.
Yeah, good question. Yeah, so, well, there's the technology of evolving these molecules, and then there's what happens when they actually evolve themselves. So when we evolve them now, we put a lot of pressure on getting it right, on fidelity. The selection scheme to answer the question about how do you pick which are the winning molecules, what we're doing now to pick the winning molecules is the molecules have to synthesize an RNA enzyme. And if they don't synthesize it accurately, the RNA enzyme doesn't function. And we choose an RNA enzyme for them to synthesize that is got a lot of potential for this wobble mistake, all right? But in general, if they don't get the letters right, they don't make a functional RNA enzyme. And the selection in that case is they have to make a functional RNA enzyme that its function rewards them with replication, all right? So that's operationally how we're evolving them to make less mistakes. But it's my view, and I think that, there's a lot of theory to back this up in observation in biology, that if we got to the point where there was self-sustained evolution, it was just going on its own, right? We weren't directing the evolution, but it just was serial transfer experiments, right? There would be a selective advantage in improving fidelity, because if you had higher fidelity, you could have a longer genome, right? Because the more accurate you are, the more information you can maintain. And if you had a longer genome, you could then use the information content of that longer genome to invent better functions, more selectively advantageous functions, including better fidelity, which would give you a longer genome, and so on. So that's a bootstrap process that I imagine, and Manfred Eigen was the first to spell this out mathematically, that once the ball got rolling, there would be selective advantage in progressively improving fidelity to improve genome length, to improve fidelity, to improve genome length, and of course, at the same time, invent other adaptive functions. So that's a kind of two-part answer.
So all right, one more, and then I'm going to take the last question. Or maybe, no.
Elena Keys, I'm a guest. And my question is, okay, better?
Good.
Great. Jerry, what do you see as major predicament or an obstacle in your research? It's one question. And the second question is, have you tried to expose this system, self-replicating system, to an environment that is already organized, say, create epigenetics?
Wow, yeah.
First introduction to what epigenetics is.
Okay.
Epigenetical factors.
Okay, two good questions. So, what is the obstacle that I see ahead? Frankly, I don't see an obstacle anymore. I really think that where we got to in 2016 was a kind of breakthrough, where we could actually copy the copies and get through sequences that we could never get through before. But I don't know how long it's going to take from here, how many more rounds of tests of evolution, how many more postdocs, how many more discussions in the lab about this, that, and the other thing. But I don't actually see an obstacle anymore. But until evolution does its work for us, we don't know. Now, the question about intermingling this with biological entities, we haven't done that. I mean, there's been some applications, as I mentioned, but we haven't done that, but with one exception that's kind of, I don't know, creepy maybe. But when I was telling you about the D and the L, the two replicators, all right, one of the applications for the reason we wanted to have an L replicator, I mean, so why even do that experiment? If the D replicator behaves with D substrates and replicates, of course the L replicator has to do the same. If it didn't, that would be big news. It'd be a violation of symmetry laws of physics, okay? So why even do the experiment? Well, because we thought it'd be cool to have an L replicator, because we've used our replicator as a molecular diagnostic where it only replicates if there's a particular target molecule in the mixture, a target metabolite or a drug that we're trying to measure its concentration. And if the target molecule is present, the replicator replicates. This picture is actually from that kind of work. This is also in a different context, but using these kind of analytical tools, you can measure the concentration of the analyte based on the quantitative replication profile of the replicator. Okay, but what if you're trying to measure a protein in human serum using this D, right-handed biological RNA replicator? Well, that doesn't work because human serum has ribonucleases in it. It has enzymes that degrade RNA. And if you remove the proteins from the serum so that you don't degrade the RNA, well, then there's no proteins in the serum to measure, right? You could try to inhibit the ribonuclease, but we thought if we make the whole thing L, then there's nothing in biology that can touch it. And this is the creepy part, because we've replicated the left-handed RNA in human serum. But not to worry, because the building blocks, the A and the B, left-handed, are not in your serum. On that note.
Well, I have a question. I have a last question. This is kind of a related question. But I think prebiotically in the experiments that have been done, you don't necessarily end up with only the standard ribonucleotides. You have modified ribonucleotides, and of course, those are found in tRNA in a zoo of a variety. So have you looked at, like, just starting with instead of just the normal ones, starting with a whole bunch of different ones and seeing?
Yes, but not for the camera. So,
okay.
Yeah, so very recently we've looked at, as I showed you on that toward the end, RNA makes DNA, DNA can get back to RNA, DNA can make DNA. What about other things? So we have such powerful polymerases now that we can actually walk them out of normal nucleic acids and into the kinds of things you're talking about. So we do need to complete that work and get it out there. But that one I left out for today. It was a little too complicated.
Right.
But
Well,
good question, though.
Thank you very much. And before you go, I want to give you a token of our appreciation.
Thank you, sir. Appreciate it.
This is, of course, the framed copy of the
Very-
announcement of your lecture.
Very nice.
Signed by all of the members of the General Committee on behalf of the membership. And
Thank you.
that's not all. There is also
I already had dinner. What else is there?
a signed copy of
Volume
Volume
1, yes.
of the PSW Bulletin, which explains the founding, the purpose,
Very nice.
and lays out who some of the original founders were, including the original president, Joseph Henry.
Wow. Yeah, that's very nice. On the original vellum, too. This is fantastic.
Yeah.
Thank you so much. Thanks,
Thank
Larry.
you. Bye-bye. So you can just,
All right.
So before we go, there are, of course, the housekeeping items to attend to. First and foremost, thank you to all of our members and sponsors and supporters and guests for coming here tonight and for supporting PSW. PSW depends on an enthusiastic, active, and capable membership. And without all of you, PSW would not continue and would not be able to grow. If you're not a member, please join. You can apply for membership on the PSW website. It's really not hard. Just, I don't know why, let's see. Yay. So just go to the home page, and there's a little button called Membership. And that will bring up this form, which you fill out. When you get down to the bottom, there's going to be a little Submit sign, which will take, There's a missing page here. It will take you to the payment page. And you can pay with a credit card. You don't have to pay by PayPal. And you just fill out the payment page in the usual fashion. And in due course, you will be admitted to membership unless you think that we're all about philosophy, in which case you'll get a phone call. And we'll try to explain that we're actually about science, not philosophy per se. There are no IQ tests, and there are no barriers to jump over. Membership is welcome to all who are interested in science. You can use that page to donate, not the membership page, but there is a button that will allow you to give donations on the home page. If you have any questions about it, please see our membership chairman, James, or our corresponding secretary, Robin Taylor. Or I guess our treasurer, John Ingersoll, or me, but preferably them. Okay, the next lecture. The speaker will be the 2,384th meeting. Yeah, on 1st December 2017, right here in the Powell Auditorium. And the speaker will be Nima Arkani Hamad, who is a professor at the Institute for Advanced Study in Princeton and also the A.D. White Professor at Large at Cornell University. He will be speaking on the doom of space-time, why it must dissolve into more fundamental structures. Of course, Nima is a theoretical physicist, not a video game author, and the talk will be about reality, perhaps including alternatives and alternative dimensions. In fact, Nima is one of the world's leading theoretical physicists, and we're really very fortunate to have him coming here to speak. He's done exceptional work in mathematical physics and the physics of dimensionality. He is also currently leading an effort to build a Higgs factory in China, which seems to be the only nation with the money and the will to undertake the construction of a collider 50 kilometers in circumference, which is twice the size of the Large Hadron Collider in Geneva. Last year, when Nima's colleague David Kaplan spoke at PSW, before David's lecture, we played an excellent film that David himself produced about theoretical physicists. Yes, believe it or not, it is an interesting and good film, including both Nima and David, the LHC and the discovery of the Higgs particle. It is called Particle Fever, and I would like a show of hands to see if there is enough interest to warrant rescreening the film before Nima's talk next month. So if you might be interested to see the film before his talk, please raise your hand. Now, you all promised to show up, right? All right, so it looks like there is sufficient interest, so We will arrange to show the film before the talk, and we will announce it on Twitter and other social media, PSW, and also send out an announcement by email. The last lecture of the year will be on December 15th, and the speaker will be Douglas Smith of Duke University, and his topics will be metamaterials. That was the old title. The new title is Cloaking Devices and Perfect Lenses, Now Structured Materials with Impossible Properties. Doug is a leader in the field of nanostructured materials that have properties that are impossible for conventional materials. Among these are unique optical properties of lossless refraction, frozen light, and cloaking devices. Technologies based on such materials are going to advance many other areas of science and enable nearly magical materials and devices in the marketplace. The spring 2018 lecture series is under construction, partially set, and should be done soon. As of now, the speakers will be Aviv Regev, who's at MIT, is a Howard Hughes medical investigator, and has an appointment at the Broad Institute. And her topic is going to be the catalog and diversity of cells. Aviv is a fantastic young researcher who has developed methods to do whole genome expression profiling on single cells, and to do it on large numbers of cells in parallel. Her work is rewriting our understanding of how many different kinds of cells there really are, and how much a given type of cells vary in its gene expression profile, as well as other characteristics. It will be a very interesting talk, well worth hearing. On February 9th, we have Christopher Ralston from George Washington University speaking about faking cultural heritage. Chris is a brilliant linguist capable of rubbing a few feathers with the facts. He likely will be telling us about the trade in antiquities and in forgeries, including how we detect them, and about the ways in which antiquities frauds impact scholarship, and when they have religious significance, communities of believers. Not always what you might expect. Religious communities sometimes know that a sacred object is not what it was originally believed to be, but nonetheless continue to revere it for what it symbolizes. On February 23rd, we have Tom Zurbuchen of NASA, and we have yet to determine his exact topic. On March 23rd, we have Andrew Knoll of Harvard speaking about life on Earth, the deep history. He will be talking about identifying and studying fossils that constitute the remains of the earliest living things. This is a subtle and challenging art, and I'm sure you will find it interesting to learn about these far distant relatives. But are they? And whether and where we might find the residua of other earlier forms of life on the planet. How did it come about? How did it change into us? If you want to know more, attend Andrew's lecture. On April 6th, we have Jordi Puig-Suari from Cal Polytech on CubeSats, which he co-invented. CubeSats provide a standardized and miniature platform to space that is allowing many more players to get into orbit for prices orders of magnitude less than those of conventional satellites. Dozens of CubeSats can be launched in the space left over on a conventional mission carrying other typical payloads, opening operations in space to a much broader range of applications, and best of all, creative minds. And conventional satellite technology is moving in the direction that is pioneered by CubeSats. And finally, as of now, on May 18th, we are planning to have a Cassini special for the Joseph Henry Lecture. I'm working hard to put together a set of several presentations on Cassini by scientists who brought this fantastic space vehicle into being, got it to Saturn, oversaw its navigational gymnastics to see the giant gas giant, its rings and moons from many angles, and who have interpreted the vast, rich ocean of data that Cassini sent back to us during its many years inhabiting the Saturnian system. Truly one of the most successful of all deep space exploration missions, Cassini's legacy is to have changed virtually completely our understanding of this complex system and its wonderfully diverse and individually complex moons, from those small rock rocks causing wakes in Saturn's rings, to the water world Enceladus and its exquisite ice geysers, to the alluring ocean world Europa and its fissured frozen crust, with the promise of warm liquid border below, possibly harboring at least a microbial form of life. We hope this will be a visually stunning, scientifically compelling, and personally engaging celebration of the Cassini team's tremendous achievement and the stupendous gifts of data, knowledge, and insight that they and Cassini itself have given to us. For now, thanks for joining us. The social hour ends at 10:30, after which PSW members and their guests meet at the Fairfax Hotel Lounge just across Mass Avenue. If you want to join us, please speak with Corresponding Secretary Robin Taylor, Vice President Lloyd Mitchell, please use the side entrance to exit the building, and I will now accept a motion for adjournment of the 2383rd meeting of the Society to the Social Hour. Second? All members in favor of adjournment, say aye. All members opposed? The meeting is adjourned to the social hour.